similar stories. But I think she has a very unique name for her particular mycobacteriophage, and I'll let her explain that. So with that, I'll give it to Lindsey.
Okay.
Hi, I'm Laura Lindsey Jackson, and I'm doing my presentation on the extraction, isolation, and sequence analysis of Mycobacteriophage Kalitakesapough and Eros. Alright, so for starters, what is a bacteriophage? A bacteriophage is a virus that infects a bacterium and uses it as a host for replication and function. When most people hear bacteriophage or anything about viruses, they think crazy sci-fi zombie stories, which is not the case for any viruses that we know of so far. But on a smaller scale, that's kind of true. A virus infects a bacterium and a bacteriophage, much like a zombie will bite another human and turn into a zombie. So the new bacteriophage is kind of like the virus's zombie. And this is a picture of a bacteriophage on the top right. And the bottom is a picture of the lytic and lysogenic cycle of viruses. So that's kind of how they enter their host, and they either integrate themselves into their genome and replicate, or they kind of make it explode and spread throughout your body, or wherever it is. Next, what is a mycobacteriophage? Why mycobacteriophage? Well, the prefix myco, it is related to mycobacterium, and mycobacterium is the type of bacterium that we used as the host for this virus. And although many of y'all aren't familiar with the genus Mycobacterium, it is actually responsible for a few common diseases such as leprosy and tuberculosis. So why are these important? Bacteriophages, they have a biological and a medical importance. Primarily they're important biologically because in the past 10 or 15 years, a lot more research has been put into discovering new mycobacteriophages and understanding them more in depth, and also understanding them geographically, which is kind of related to our research. On the bottom, you can see a picture from a mycobacteriophage database. This is a picture of the United States of various mycobacteriophages that have been submitted to this database. So this is one way we can kind of look at them geographically and see how they're related, or if they're related. And then also they have medical importance, because if you turn on the news today, you've been to the doctor, you read a magazine, you probably heard the huge hype about antibiotic bacteria that are growing out of control. MRSA, for example, methicillin-resistant Staph aureus. It's a bacteria that has grown resistant to many different types of antibiotics. And with that being said, they're looking for new ways to treat people other than antibiotics. And one way is called phage therapy. And it's the use of lytic bacteriophages to fight pathogenic bacterial infections. This phage therapy was actually used quite frequently in Europe before the invention of penicillin, but since penicillin was invented and other antibiotics have been derived, it's kind of been pushed off to the side because penicillin is simply easier, it's cheaper, and it's quicker. And naturally, that's what they're going to use. So, but recently there has been more research with phage therapy and looking into its therapeutic uses. And the more we understand biologically about bacteriophages, we can understand more about them medically. The objective of this experiment is to isolate a novel mycobacteriophage from a soil sample, and this is a class-wide experiment. And so everyone in the class wanted to isolate a novel mycobacteriophage, and then it was going to be sent off for analysis at the University of Pittsburgh and eventually entered into a mycobacteriophage database. And this is a picture of a mycobacteriophage named Pixie that has been annotated. Alright, so primarily we had, as a class, everyone collected soil samples, and we were given a week to do so, and naturally I procrastinated until the morning of my class, my 7:30 molecular biology class. At 6:45 in the morning, I walk outside my house to collect my samples. I was originally going to collect them from this area right here, my side yard, where my dog Callie, she frequently poops, and I was going to collect it from there, but I come outside and it's raining. And the one thing Dr. Murdoch said is, do not collect them after it rains. So I'm thinking to myself, if I'm soil-infested or poop-infested soil, where am I going to go in the event of a rainstorm? And so I ended up collecting the soil samples from right here, because the runoff from, this is kind of a hill, you can't really see that well from the picture, but it's a slight incline, so the soil came from here and down into here. Figured I might get something out of there, and turns out we did. First, we had to filter out the virus from the soil sample because we don't want the soil particulates in our culture. So using a syringe type tool, we squeezed out the virus kind of liquid from the soil sample. That's what this picture is demonstrating right here. And we used the enrichment method to enrich the virus. And enrichment is basically a fancy way for saying infecting. So we put the bacteria and the virus together and hope that the virus would infect the bacteria. And to do this, we use Mycobacterium smegmatis, which is right there. And it's actually similar to Mycobacterium tuberculosis, which is the pathogenic mycobacterium that causes tuberculosis. And the enrichment method is one that's been used in many different experiments, and it's been proven to be very effective, so that's why we chose to go with that one. Next, we had to take the sample and we had to plate it on LB agar. And this is actually a picture of the first plate. So once I realized that we had growth, and although this is a little counterintuitive, the growth is actually the plaques of clearance. So where you see little clear spots is where we had growth, because that's the virus eating up the food that we gave them in the enrichment. So once we realized that we had growth, we had to shriek for isolation, because we had uniquely, we had two different morphologies on our plate, which means that the virus is showing up in two different kind of disguises. And it could have been two viruses, or it could have been the same virus that had multiple morphologies, but we had to choose one morphology and go with that. So we tried to isolate one morphology and stuck with that for the rest of the experiment. Next, once we had a virus, a mycobacteriophage, isolated, we had to do DNA isolation, and we did this using Promega DNA Wizard, and we basically just took the DNA out of the virus, and then we ran RFLP, which is restriction fragment length polymorphism, and a long word, but really all it means is a DNA fingerprint. So we kind of cut up the DNA and looked at its parts in order to understand the DNA as a whole. And this is a picture of the gel that we ran for the DNA extraction for the RFLP, and there's a key of the different lanes of the gel, what's located in each lane. All right, so after we extracted DNA and everyone in the class, a couple people got DNA extracted, we all sent off our samples to the University of Pittsburgh for analysis. Well, we sent off a couple samples from our class, and unfortunately mine wasn't chosen for analysis. So for my results and discussion section, I'm going to do it on another virus from the class named another mycobacteriophage from the class named Eros. And Eros is part of this Mycobacteriophages family, which is a family of double-stranded DNA viruses. And viruses can be classified in a few different ways. They can be enveloped or non-enveloped. In our case, it is not enveloped. And they can also be classified by the shape of their head. And in this case, it's a icosahedral, which is kind of a funky geometric figure. And then also we found out that it has a non-contractile tail. And this is a TEM picture of the mycobacteriophage on the top right. And this mycobacteriophage Eros was actually collected right behind here. So if you got up and looked out the window, you'd see the area of collection right behind the library at the very back of the parking lot. All right, and after annotating the genome, this is a screenshot of the annotated genome. We discovered that it has around 50,000 base pairs in its genome. It also contains about 87 protein encoding genes. And one thing that was really interesting about this mycobacteriophage Eros is that when we put it into the system that compares all these different mycobacteriophages, it came up with a 99% homology with peaches. And homology means basically what they have in common. And so it has 99% of its genome it has in common with this other mycobacteriophage, peaches, which is interesting. Another thing that's interesting is peaches and our mycobacteriophage fall into the same cluster. It's the A4 cluster. And they actually have only been located so far in the eastern United States. So this mycobacteriophage database project is worldwide. China, Europe, all these different places submit mycobacteriophages that they found and isolated. And A4 clusters are only found in the eastern United States. So this is something very interesting that we can further look into for research. And that's a picture of the various A4 clusters that are in the eastern United States. And lastly, we found out two really interesting things that we could take from this research experiment. One is that this genome is extremely dynamic, which means that it's constantly changing. It's constantly taking genes from not only other viruses that it comes in contact with, but also the genome that it inserted itself in. So it's taking genes from the bacteria that we infected with, the mycobacterium. So that's really interesting. And also, our final conclusion is, and something we could look into for further research, is that I said mycobacteriophage infection as benchmark. So all of these mycobacteriophages, they have something in common, and that is that they infect mycobacterium. And from there, from comparing these together, we can see how they differ. And maybe this is kind of a picture of a family tree of all of the different mycobacteriophages, basically viruses that infect mycobacterium, and Eros is right here. So from this experiment, we can kind of see what Eros has in common with all of these other mycobacteriophages. And lastly, I would like to thank Dr. Murdoch for making this class a research opportunity as much as anything, which is really important for people of my age. And also wanted to thank the Howard Hughes Medical Institute and the University of Pittsburgh for helping with the analysis and whatnot. And also wanted to thank the class for everyone's participation in collecting soil samples, because this was a joint effort, and also the biology department for letting us use their facilities and for using their equipment.